Diatomáceas.
Olá, pessoal. Eu acho que já são 14 horas e 4 minutos. A gente já poderia iniciar os nossos trabalhos. Então, inicialmente eu vou começar falando em português, depois eu peço licença para falar em inglês, e peço a licença ainda maior para ler uma parte em inglês para não cometer deslizes muito absurdos, porque há algum tempo já que estou longe do inglês, então não vou cometer essas gafes com o palestrante internacional, né? Mas, então, primeiramente, eu gostaria de agradecer a todos, pessoal. Essa é uma iniciativa, eu acho que importante para nós trabalharmos com a questão da rede, certo? E nesse processo, a gente acaba executando alguns dos objetivos da nossa rede de diatomólogos, que é interagir o pessoal através de encontros e através de eventos como esse. Enquanto o encontro de diatomólogos, né, diatomologia é algo mais formal, o congresso de botânica, nosso objetivo aqui foi fazer uma coisa bem informal, tranquila, para que vocês consigam conversar com os palestrantes, tratar com eles diretamente sobre as questões importantes e estabelecer até mesmo uma rede de contatos aí com eles, né? Hoje, nosso moderador será o Maylor. Para quem não conhece o Maylor, ele foi graduando na UniOeste, né, Maylor? Tendo contato lá com a Margot, com a professora Norma, né? Atualmente, ele é doutorando em Biologia Evolutiva e Ecologia, né, pela Universidade do Colorado. E também ele atua lá como professor assistente da disciplina de Biologia e Sociedade desde 2022. Ele é licenciado em Ciências Biológicas e mestre em Conservação e Manejo de Recursos Naturais pela Universidade Estadual do Oeste do Paraná, UniOeste. E durante sua formação ele desenvolveu pesquisas taxonômicas, ecológicas, com diatomáceas, tanto fitoplânctônicas quanto perifíticas, mas com foco lá na microbacia do rio Cascavel, no Paraná. Atualmente, o trabalho de doutorado dele, olha que legal, integra taxonomia sistemática, genômica e filogenética de diatomáceas, com ênfase no genoma do cloroplasto de Rhopalodiales e as cianobactérias endossimbióticas ali com esses grupos, né, que a gente sabe. E aí ele vai atuar como mediador, então, da nossa sessão e vai explicar, inclusive, qual que vai ser a dinâmica que a gente vai atuar. Agora eu peço licença a todos, então, para fazer uma apresentação em inglês, né? Inclusive apresentando para o Patrick a nossa rede. Bom, good afternoon, everyone. It's a great pleasure to welcome you to the first edition of Papo Diatomáceas, né? Before we begin, I would like to briefly introduce the Brazilian Network of Diatomologists, which is the initiative behind this event. The network was created with the goal of connecting researchers working with diatoms in Brazil, promoting integration across different areas such as taxonomy, ecology, paleolimnology, and molecular biology, while also strengthening collaborations, technology exchange, and the training of new specialists. The idea behind the network is quite simple, yet very important, to create an open, collaborative, and ongoing space for dialogue, a space where we can share scientific advances, discuss challenges, including conceptual and methodological ones, and also bring together students, researchers, and anyone interested in this important group of organisms. Within this context, Papo Diatomáceas was created as an initiative for scientific outreach and integration. It is a more informal and accessible space without losing scientific rigor, where we invited researchers to discuss current and relevant topics in the field. Today, we are very pleased to welcome Professor Patrick Kociolek, who kindly accepted our invitation to speak about classification of diatoms, phylogeny, reshaping systematic. For this season, we are also glad to have Mailer Amaral, who will act as the moderator, helping to guide the discussion and facilitate the questions and answers section. Mailer is a PhD candidate in ecology and evolutionary biology at University of Colorado Boulder, where he also been serving as a teaching assistant in the course of biology and society since 2022. He holds a bachelor's degree in biological science and a master's degree in conservation natural research management from the State University of Western Paraná. During his academic training, he conducted taxonomic and ecological research on phytoplanktonic and periphytic diatoms, with focus on the Cascavel River in Paraná, Brazil. Currently, his doctoral research integrates taxonomy, systematics, genomics, and phylogenetics of diatoms, especially emphasizing in Rhopalodiales. Finally, I would like to sincerely thank Patrick for his availability, as well as Mailer for moderating this section, and all of you, todos vocês, for your presence and interest. Without further ado, I will now hand it over to Mailer to introduce the event and walk us through this format. Okay?
Ok. Primeiro de tudo, obrigado, Wellington, pela apresentação. Pat, I would like to ask your permission for a few more minutes just to make this overall presentation in Portuguese very briefly. And if I'm not mistaken, most of the people here are Brazilian, so I think Portuguese is the way to go for just a few words.
Sure.
Ok, so sejam todos muito bem-vindos, Como o Wellington falou, a nossa primeira reunião online da Diatom BR, ou da nossa Rede Brasileira de Diatomólogos, cujos encontros serão realizados, então, sob o nome de Papo Diatomáceas, literalmente um papo. Hoje contamos com a presença do professor Dr. Patrick Kociolek, a quem nós agradecemos por aceitar o convite e também compartilhar a sua experiência conosco. Dando início ao nosso evento, então, nós estaremos reunidos aqui por cerca de 2 horas, então vamos ter um bom tempo para bater um papo com o Kociolek. Para manter esse formato mais informal, de papo mesmo, o Dr. Kociolek vai começar com a sua apresentação, mas os participantes são encorajados a fazer perguntas a qualquer momento da apresentação. Então, seja levantando a mãozinha no chat, caso vocês se sintam confortáveis em fazer a pergunta diretamente ao palestrante, eu posso deixar ele sabendo que vocês têm uma pergunta, ou também mandando as suas perguntas diretamente no chat. Eu vou estar mediando a sessão e também posso ajudar como tradutor, se as pessoas se sentirem mais confortáveis em fazer as perguntas em português, sem problema nenhum. Fiquem à vontade, não se sintam acanhados para participar, eu vou fazer o meu melhor para transmitir as perguntas para o palestrante. So now, I will switch our conversation to English. It is my honor and great pleasure to introduce our speaker today, Professor Patrick Kociolek, who is also my advisor. Pat is a leading expert in diatom taxonomy, in systematics, in evolution, with a long-standing career dedicated to the study of and classification of microbial diversity. Throughout his career, he has held major leadership roles in natural history museums, including serving as an executive director of the California Academy of Sciences in San Francisco, and also as a director of the University of Colorado Museum of Natural History. We sincerely appreciate your time and willingness, Pat, to share your work with us. I'm sure everyone is excited to hear your talk and also to engage with this discussion. And so today's talk, again, is titled Classification of Diatoms, Phylogeny, Reshaping Systematics. based on one of your recent publications. So questions will be welcome throughout the talk via the chat box. People can raise their hands, so either in Portuguese or English, and I'll bring the questions up as they come and as they show up. Without further ado, so the floor is all yours, Pat.
Perfect. Well, Wellington and Mailer, thank you so much and for the invitation, and thanks for everyone for joining. And so I just want to say a few words here. You know, I think as I understood, our goals today is really just about communication. And I hope that all of us can feel comfortable expressing our points of view. Don't let hierarchy get in the way. If you're a young person and you have questions, don't look over to see what your professor's thinking or, you know, looking at you. Just ask the question. You know, we want to make sure that this is a very open forum. My job, of course, is to hopefully speak clearly and deliberately. Since Portuguese is not my native language, that might be difficult for me. And so I'll do my best, and I hope that I can be a good communicator about the ideas that I want to share with you. For you, however, I think that there's some responsibility. One is to let me know if I'm doing a good job, and that might be, you know, I'm excited about this stuff. I really am super excited about this work, and I'm on my third cup of coffee, and so I could start speaking very quickly. And or maybe I'm just not communicating well. So if you are having a hard time understanding, just let us know. There's no problem. I can easily slow down a little bit. And if you have a question during the presentation, I'm happy if you want to raise your hand and ask for a point of clarification. The goal here again is to make sure that we're all communicating. So if English is not your first language, ask a question in Portuguese, and Myler and others will help communicate that to me and help communicate my answer back to you. So let's make this a great opportunity for communication, and let's chat. So here we go. Here's how I'm going to organize the talk. I'm going to talk about what is a classification system? What are we doing when we think about making formal classifications? I'll talk a little bit about past efforts to create classification systems for diatoms. I'll talk about the approach that we took in developing this new classification system. Talk a little bit about the new classification and how it relates to past efforts. And then I'll talk a little bit about some of the challenges of this work, as well as potential next steps. So that's how I hope to keep my thought process in line as we chat about diatom classifications. So what I'm going to present ultimately is a new, what we would say, natural classification of the diatoms. So what is classification? What are we doing? It's a way to organize information about relationships based on our understanding of systematics and phylogeny. We want our formal classification systems to be natural. That is, to reflect evolutionary relationships of taxa. Now, there's all kinds of other classification systems one can use. They can be convenient, they can be easy, but for a formal classification system, we want one that reflects the evolutionary history of the group. And of course, in a formal system, we in biology use a Linnaean classification system with a set of in a hierarchy that goes from kingdom, division, class, order, family, genus, and species. So we want to use that structure. And we just have to realize that sometimes things might be classified together that don't look similar to one another. And that is, but there ought to be some basis for that, that decision, and how we can make the basis for our decisions most transparent and most reproducible as scientists is an important goal of ours. So we could think of classifications as natural ones. We are the ones that reflect evolutionary relationships, and here are some examples of natural classifications. Diatoms are a natural group. the raphid diatoms within the group as a natural group. And we could have classifications for ease or convenience. We could say things that we want to talk about the plankton, or the things that fly, or things that are nocturnal. As it turns out, things that have amphoroid symmetry are all groups, but they're not natural groups. And so there might be a reason to talk about plankton or amphoroid diatoms, but we wouldn't formalize them into a natural classification system. So a concept that we use in this regard is the idea of monophyletic groups. So monophyletic groups include an ancestor and all of its descendants, and that's as opposed to paraphyletic groups that might include only some of the descendants of an ancestor. So highlighted in red here on the right-hand side of the screen are different groups within a phylogeny in a tree that we might identify as monophyletic groups. It includes the ancestor and all of the descendants. Where a paraphyletic group might have an ancestor but include only some of the descendants. And so we want to base our classifications on those groups that are monophyletic. It's an important concept in systematics, and one that drives our work in developing this new classification for the diatoms. The wonderful thing about monophyly is that it can be predictive by identifying an ancestor and all of its descendants. We could look for shared evolutionary features and say things that are together in this monophyletic group probably have similar responses to ecology, perhaps, or other interpretive materials that we can overlay on a monophyletic group. So these are the kinds of groups that we want to give names to, just monophyletic groups. Now, some other concepts that we might want to talk about briefly in the idea of identifying monophyletic groups is that we might want to look for homologies. These are characters that are shared between groups that are due to shared ancestry. So it's these kinds of characters then that we want to base our classification systems on. So we can give an example for the Bacillariales in the genus Nitsia and its relatives. We could say that a keeled raphy or canal raphy is a homologue across all of the members of the Bacillariales. And so that would be one type of homology. Homoplasy is that things that may have what look to be similar characters, but they're not derived from a common ancestor. So a lot of times this is the result of convergent evolution, where two very different lineages have arrived at the same answer or the same feature. And we don't want to use homoplasious characters. We want to use homologous characters to develop our classification systems. So we could say, for example, in terms of homoplasy, we could use the term wings to identify the things that are on insects, birds, and some mammals, but they're not derived from the same anatomy. They've been independently evolved for flight, and we wouldn't put insects, birds, and flying mammals together based on the feature of wings. So that's the idea. So we have these two different types of features, and we want to use homologous characters. So those are important concepts in evolutionary biology and systematics and phylogenetics. And just two other terms that might be useful for us is the term synapomorphy. And in both of these cases, both of these terms, features are shared. But in synapomorphies, the features that are shared are derived features. They're evolutionarily derived. And through decades of work in systematics, it's been identified that synapomorphies are the types of features that help diagnose lineages and help identify natural groups. That's in opposition to simplesiomorphies, that are features that are shared but primitive. And so for an example, we might say that these diatoms have silica in their cell walls, but the presence of silica in a cell wall doesn't help you identify the difference between the naviculales and the surreallales. It's a feature that's primitive to the diatoms and primitive to groups like that. So we want to base the features on or the classification system on features that are synapomorphies. How are we doing so far? Can you hear me okay? Is my pace of speech okay for everybody? Perfect. Thank you for that feedback. I don't know if you didn't agree, instead of a thumb, do they put up a middle finger or something like that? I don't know how that might work, but anyway, we'll take it that things are good here. So we'll keep move along then. So of course, the classification system I'm going to talk about is not the first one for diatoms. There's a long history of researchers looking at diatoms and coming up with ways to group them in classifications, both formal and informal. And the different types of classifications that people have developed, here's how I would identify them. Some of the classifications were made with observational data. The individual scientists or group of scientists might say what they think of those data are important, and then they would base a classification system on those important characteristics. Other approaches might be people who have thought that a character might be a good character. Sometimes the Raphy is thought to be a good character in classification. Sometimes people group things together or have grouped things together because they're complex. The idea is like, man, that is such a complex feature. There's no possible way that could have been derived separately. And so we'll put things together, we'll group things together based on these very complex features. And then some people might say, we think that there are characters that are very conservative, that don't change much at all. And so based on these conservative features, we'll come up with classification systems. So then there's been people who have developed trees. They might draw an evolutionary tree, but how the tree was developed is not clear. There's no formal analysis. You can't reproduce it. You can't go back to what they looked at and create that tree again. And no use of monophyly to identify the groups. And then there's others where there's been a tree, there's been formal analysis. Many of these have been molecularly, the data have been on molecular data, but there's been no adoption for monophyly. And I'm going to talk about each of these different types. And then finally, this project has trees, it has formal analysis, it's mostly based on molecular data, and we have used monophyly as the basis for establishing the classification system that we did. So just in terms of history, here was a group that was called the Arcuatiae. It was a family called the Eunochiaceae, and the light micrographs here are pictures of diatoms that were all put into that group by Agard and followed by Kutzing later. So they thought the good character was the shape, that shape and symmetry were really important. And so things that we know of as Eunotia, Epithemia, and Cymbella were all included in the same grouping based on this idea that shape was important. Other people look to not the cell wall features, but the features of living diatoms to create classification systems. So Petit and Van Herk and Pfitzer all use the features of the types of chloroplasts. As you know, some diatoms have many small discoid chloroplasts, and others have just a few very large plate-like chloroplasts, relatively large plate-like chloroplasts. And some people thought that was a super important character, and so they created a classification system based on the types of chloroplasts that were present in the diatoms. Others thought that movement was a really important characteristic, and Mereshkovsky, who is a very important person in biology generally, he's the person who came up with the theory of endosymbiosis. He believed that movement, the ability to move or not having the ability to move, was an important characteristic, and created not only a tree, but created a system that was based on this feature. Perhaps the most famous and most long-lived classification system was first produced by Schuett and later by Karsten, that identified two great groups of diatoms. And maybe the staying power, the longevity of this system is that they integrated a lot of different information in to put this together. So they identified centric diatoms having features that of the glass cell walls of expressing radial symmetry. But in their system, centric diatoms are also oogamous. They produce sperm and eggs during the sexual process. And they mostly have many small discoid chromatophores. And that's as opposed to the pinnate diatoms, whose symmetry is bilateral. Most pinnate diatoms have isogamy, where the gametes are basically morphologically and many times physiologically similar, indistinguishable, and that they have a few large band-shaped chloroplasts. And that system by Schuett lasted for about 100 years. And it was ultimately surpassed or taken up by a system that was developed by H.L. Smith, but was later adopted by Round et al. And that's the idea that there are three great groups: the centrics, the raphids, and the araphids. Some other systems that you may or may not know about, this guy Bessey was one that decided he saw centric in pinnates, but he basically then produced a classification system that was based on the idea that filamentous diatoms were primitive and individual diatoms were derived. One of the interesting things about this system by Bessey, he identified that the Surrelaceae here are quite distinct from the Nitsioid diatoms of the Bacillariaceae here, even though they have a very similar raphe system. He's probably the first one to identify that difference, and that difference has been suggested recently in molecular data. Now, a very influential approach was Simonsen, who created this system that identified centrics and pinnates. And then he had this tree that he developed, but not clear on what that's based on or how one would reproduce that tree. But he then identified a number of groups within those two major divisions, or in this case, orders, almost none of them monophyletic. And as I mentioned, Round, Crawford, and Mann came up with this system that they borrowed from H.L. Smith, but not clear on what those categories are based on in terms of data or reproducing it. This is Smith's original breakdown of those groups. So you could ask yourself, for all of these different classification systems, how do they come up with it? You know, how do they come up with it? What approach did they use? What about their results and their conclusions? Could you reproduce those based on the work that they did? And if not, why not? And if not, is it science? Is the work we do reproducible? And I think this is just for the younger members of this audience, please keep in mind that it's super important as a scientist to make sure that your work can be reproduced. Somebody ought to be able to read your paper, get back to your data, analyze your data in the way that you did, and come up with the same answer. And if they can't, that is a big challenge. And right now in science, as you may know, that is a huge issue. And we could talk about why that might be. So in the approach by Medel and Kaczmarska, they used molecular data and they made an analysis and they made a tree, but instead of identifying in their classification system monophyletic groups, they created clades. They identified grades of organization rather than monophyletic clades. So in the kind of pink section or peach section there, they identified radiocentrics. In that green section, they identified non-radiocentrics and identified a group. And then in parts of the blue there, they identified, not sure why this is not going forward, but they identified araphid diatoms that are all over that tree. So hold on, oh, sorry, let's go back there. So they identified this radiocentric group, the non-radiocentric group, and all of those things where the arrows are araphid diatoms, and they put all of those in the same category as well. So they identified grades of organization rather than clades of relationships. So at the bottom here, this is basically taking what Medel and Kaczmarska had as their answer and the groups that they identified. And I just want to show you kind of how these different past classifications would match up to what we know or we think of as the set of relationships today. So you can see down here, different lineages of what was called radiocentrics, non-radiocentrics, araphids, and then this big group of raphids here. So if you took Schuetz's classification and said, here's centrics and pennates, the pennate diatoms are monophyletic, but none of the centrics are. And if you took Round et al.'s classification, neither centrics nor araphids are monophyletic, though raphid diatoms are. If you took Medel and Kaczmarska's group, radiocentrics are not monophyletic, non-radiocentrics are non-monophyletic, araphids are non-monophyletic, only the raphid diatoms. So you can see through time, we're actually getting worse. We went from 50% of the major groups as being monophyletic to 30% to 25%. And in a more recent classification system where two major groups were identified, radiocentrics and this thing called Basilaria phytina, only 50% of the group is monophyletic at this level of hierarchy. So we've never had a natural classification of the diatoms. We concluded from that the diatom classification system is broken, and we needed to change that. So in this paper, what we did was we borrowed a phylogeny that Andy Alverson created in a paper that was published in Proceedings of the National Academies of Sciences of the United States. And based on that tree, we then divided up that tree based on monophyly to identify a classification system, to create a classification system. So we wanted our approach to be data-driven, using both molecular and morphological sources. So we took approximately 180 genera that were part of Alverson et al.'s tree and looked at the phylogeny. We then included about another 300 genera that were not part of the original analysis, which we identified as being inferred when we placed them in a particular group. So we had some taxa that were part of the original analysis, and we put them in this classification system. Then we took another approximately 300 and identified them in the classification system as being inferred. And those inferences could come from a variety of data sources. They could be inferred from molecular work or from DNA. And so in the classification system we identified that they weren't part of the original analysis, but there is molecular data to suggest their position wherever we put it. We also did that for morphology. And if there was a synapomorphy identified for a placement of a group, we identified that in our classification by putting an S. If morphology was used and there was not a synapomorphy, we just used we put a G next to the name that says this is general morphological support, but not a formal analysis that identified a synapomorphy. So we went to the literature and looked for these relationships to help us make these assignments. And so what we were hoping to do to the greatest degree that we could is be super transparent about what we're up to, identify the references that we used to make the assignments that we made, and so that this would be as reproducible as possible. The evolutionary tree would be as reproducible as possible by the work by Alverson and all. And then our work to create the classification system would also be this high level of transparency on our part about how we made decisions and assignments. So what we found in this tree is that many of the groups that had been identified were non-monophyletic in the previous classification systems. In some cases, orders, say the navicular, orders may span between 2 and up to 9 distinct clades. So groups that were assigned to the naviculales were actually in 9 different clades and not in a monophyletic naviculales. So from this classification system, and so here's the phylogeny here. These numbers identify 10 classes that we identified for the group. This is kind of the old classification system here. These pictures just have representatives. And part of that story that Alverson et al. Told, which is super interesting one, is that for a long time, diatoms did not radiate as a group when they first evolved, but it was almost 100 million years until there was a big radiation of diatoms. But anyway, we identified 10 classes, to each of which would be monophyletic. And then we would, putting into the hierarchy of class, then we would identify orders, families, and genera. That led us to create about 20% of new taxa. And we shied away, in this approach, we shied away from creating lots of supra or sub families, orders, whatever. We didn't want to create a whole lot of other categories. We wanted to use these main Linnaean categories. And with approximately 450 taxa, it is by far the most complete classification of diatoms to date. And it's the only one that is based on these natural groups. So in this is a comparison. The phylogenies are the same on either side. It's the same phylogeny. This is the previous classification with 4 major classes. This is the classification of Cox. And here's our new classification. So you can see how these different groups are organized and how we identified them here. So let me just show you in the first 3. So let me just go back for a sec. So I'm way down here at this part of the tree. And I'm just going to show you at that part of the tree, those first branches. Here's what the previous classification had, and here's what our classification identifies. And I'll just show you how we went about putting together the groupings. So here in the first group, we identified that as a new class. We had to create some other categories here and just recognize that this work had already been done. So this genus was part of our original analysis. So we've put the genus here, and then we've said there are some folks, or there's some groups that are inferred to be here, and we identified those resources that we use to make that inference. So that's the first group. The second group is here. It's within this clade. Sorry, it's in this. Sorry, skipped one maybe. Here's the second group, the Leptocylindrociphyceae. It's got these this hierarchy and two genera that were part of the original analysis by Alverson et al. The third group, a little more complex, is the first one is this new order, and the genera that are assigned here, the one that was part of the analysis, and others that were inferred based on, in this case, morphology. A second group in there that was also this genus was part of the original analysis, and then genera that were inferred. And then finally, in this group, the genus Melocyra and Olicocyra were part of the analysis, and some other inferred genera. So that's the kind of the mechanics of how we went about organizing and assigning taxa within this new classification. Oh, here's the last little bit. Same idea. So so here's what I have here is the phylogeny that was developed by Alverson et al. And the old classification system. So I want to show you that we could look at some groups that were part of the old classification system, and just show you how some of these examples of how people have classified things in the past get expressed here. So let me just show you that. So for example, the radiocentrics, they are not a monophyletic group. It's a morphological grade. And the idea of radial symmetry is a simplesiomorphy. It's something that is at the base of the tree for diatoms. It doesn't identify a feature. And so we would not recognize radiocentrics. Given this phylogeny, and given that we want the classification to reflect the phylogeny, we would not use radial symmetry. Radial symmetry is not a feature that would be useful, and radiocentrics would not be a group we would identify. Likewise for the araphid diatoms. Here the araphid diatoms come out in about 3 different groups. They are, the araphid condition is a morphological grade. It's not a monophyletic clade. And so we wouldn't formally recognize araphid diatoms in our classification system. You may want to talk about araphid diatoms in some way, but it's not a formal group that you would recognize. You know, one feature that was thought to be conservative at one point was the number of raphe systems on a frustule. And the group, the monoraphid diatoms, those that have a single raphe system, were classified together in a single group. But it turns out representatives of the former actinianthales come in 3 different parts of the tree of life of raphid diatoms. And so we wouldn't use the monoraphid condition to group things together that have a single raphe system. We could identify the actinianthales, this group of the marine actinianthes, but other things like actinianthidium or Caryavia, we would find in other parts of this tree, and we wouldn't put these 3 genera together in a single group. Likewise, things that in the past have been called naviculales are found across the tree of life of Raphid diatoms. And therefore, because they have this kind of naviculoid symmetry or naviculoid features, we would not identify naviculales, everything with those features, as a single group. And while I use the idea that canal raphy is found in the bacillariales and is a feature that unites them as a single group, a canal raphy also occurs way over here in the diatom tree of life. And so we would not use a canal raphy to join all of these groups with the bacillariales in a single group, a single formal group based on the presence of a canal raphy. It looks like a canal raphy, even though as complex as it is, has evolved twice. And finally, you might not think of, it'd be hard to come up with a feature as more complex as an amphoroid diatom, or a diatom that has amphoroid symmetry. You know, instead of the valves being put together like a petri dish, the valves are all pushed together where you can see in one plane the valve faces of both valves of a frustule. It's a very complex set of features, facilitated by differential sized girdle bands and lots of them. But in fact, diatoms that have been put in as amphoroid diatoms seem to have evolved independently 4 different times in the Raphid tree of life. So we wouldn't identify a group of diatoms that have amphoroid symmetry together in this approach. So, there's this new classification system has some challenges to it, and we don't have synapomorphies to diagnose every lineage. It's super challenging and something we're still working on. Of course, with 450 genera, we don't have all the genera of diatoms included, so we have some work to do there. There's not a separate morphological tree. So we had this tree that I showed you was based on, 200,000 amino acids, but there's no corresponding morphological tree. A good question we could ask ourselves as a community is, why don't we have a morphological tree for diatoms, but we don't have a tree that we could then compare molecular data with morphological data? It's very difficult in this system to integrate fossils. The fossil record offers some super interesting taxa and characters that would be wonderful to have as part of our classification system. But in this molecular-based phylogeny, we don't have the benefit of those data here. And, it's very difficult. Some of these groups are very diverse in some of the groupings that we have. And we could ask ourselves a question. Well, let's ask ourselves a question. Let me see here if I can go. So this is the interactive part of the presentation. So does every taxon in a group have to possess the features that diagnose that group? What do you think? Who's willing to stand up and be counted and make a suggestion? How would you answer this question? So let's say you identify a group. You say the group is diagnosed by this feature. Does everything you want to put in that group have to have that feature? Say it in Portuguese.
I think not.
Excellent. Why? Oh, all right. So let's talk about why not. My answer is no. And here's why. Some taxa could secondarily lose features or not yet gain them, but still be part of a group. So let me show you. I'll show you an example with a genus that I'm not sure you have. It's not widely known, but it's called Dipura. And Dipura is an endemic genus from Hawaii that lives in a very, the amount of light is very low. And these things live in little cracks and crevices at the tops of caves where water drips out. And it was originally described as an araphid diatom, and you can see the light microscopy here. Here's pretty much the entire size diminution series from the initial valves down to the smallest cells. And it turns out this diatom grows well in culture. So we looked at this diatom, and because it was dividing like crazy in culture, we could look to see did a raphe occur in any part of its life history? And in fact, we could never find any hint of a raphe in this diatom. And yet, when you do a molecular analysis, so let me just identify things here. Here's quote unquote the centric diatoms out here. Here's quote unquote araphid diatoms. And from here out are pinnate diatoms. Dipura is found deep within the raphid diatom clade. It's closely related to Phylogenia and Celophora. And so even though this diatom does not have a raphe system, we would classify it amongst the raphid diatoms. We could not justify putting this diatom out here somewhere as a part of the classification system. So particularly the secondary loss is something that is certainly happens in many cases in diatoms. So we just have to be ready that even though the feature might be here of possession of a raphe system, this diatom does not have a raphe system. So for us, the next steps in this work are, this is starting to be incorporated now into Diatom Base, which is an online nomenclature for diatoms. Mike Gyrey has already introduced it into AlgaeBase, and which is good because AlgaeBase didn't even previously identify diatoms as a monophyletic group. We'd be happy if you're working on something. We'd be happy to send you an Excel file with the classification system if you want to update that. And just remember, we're trying to do science here, and we will certainly see there better be new data that may align or challenge this approach. And that's what we're trying to do. So even in our own work, we are hoping to challenge this and see how robust the classification system is. Does it do a good job of representing that phylogeny or not? And that's the next step in our work. And I hope in all of your work as well. So with that, I would be happy. I appreciate your attention, and I'd be happy to answer any questions you have. Thank you so much.
Thank you, Pat. That was great. I'm sure that people have a lot of questions, and this is the time to do it. So, if you have any questions, and se vocês tiverem alguma pergunta, quiserem fazer diretamente para o Pat ou fazer no chat, eu ajudo a traduzir. E é realmente a hora da gente bater um papo. Well, I guess you can see a lot of positive reactions, Pat.
I'm looking for the middle fingers coming up, but I haven't seen any yet.
Well, I haven't seen any as well.
That'll be the question. That'll be the question period. So I'm not sure I can see the chat. So if people are offering, if people are asking questions in the chat, I can't see it, Myler. So if you could keep an eye on that. And
yeah.
Should I stop sharing, or do you think I should leave this up in case somebody wants to refer back to a slide?
That's a really good question. It depends on how easy it is to put it back, maybe.
I think it's pretty easy.
Yeah, so maybe if you want to just stop sharing for now, and then we can focus on the questions, and then...
Perfect.
Yeah. Okay, so it seems like Kociolek has a question on the chat, and I'll read it for you. So,
Thank you.
Pat, thank you so much for taking the time to talk with us today about this important contribution to diatom classification. I wanted to ask specifically about the use of Bacillariophyta or Phyta as a phylum. So this is the term we most often use, but there seems to be a growing tendency to place all heterocont algae under Heterocontophyta. In light of recent phylogenies, do you think keeping diatoms under Bacillariophyta or Phyta could make it harder to reorganize other heterocont groups like Bolidophyceae and Dictyophyceae? How do you think we can classify diatoms while also placing them in a classification framework that reflects the phylogenetic relationships among heterocont algae?
So my understanding of that work, this kind of broader level groupings within the heteroconts, is that diatoms are a monophyletic group, and maybe so we're constrained. There's the Linnaean system creates some constraints for us in classification. And it turns out that both at this broad level that you're identifying, Kociolek, and at the level of genus, there's going to be some struggles in having enough categories to reflect the diversity of forms that we see in nature. So we took the kind of traditional approach of using the grouping that diatomists have used. It could be that at the level, and I still use the term division rather than phylum, because this is botanical nomenclature, a system of botanical nomenclature that we need to, it could be that these higher level classification groups need to be expanded. So I understand the nature of your question. And I think both at the higher levels of classification and as we continue to identify new groups at a very specific level, genera and species, the Linnaean system is going to provide some problems for us in being to recognize those groups. So there's pressure from a ton of new genera being identified, and then what kind of categories will we use to group those genera? And likewise, as there's this explosion of information about the higher level classification, Linnaean system is going to create some roadblocks for us as well. But I'm comfortable with using Bacillariophyta and hope that people will be creative about how to organize things at this higher level.
Well, he says thank you.
I see.
Yeah, it looks like we have some people with their hands raised as well. And the first one is Andrea Franco. Andrea, não sei se você prefere falar a sua pergunta e fica à vontade para ligar seu áudio também e aparecer se você quiser, ou se quiser mandar a pergunta, é totalmente...
Okay, I don't know if you can see me. I can understand, but I don't speak very well English. I work with molecular taxonomy of diatoms, but I haven't been working with few number of genes, 3 or 4 at maximum. So I would like to understand better how we can use this classification in the future. For example, I can sequence the genome of another genus that you don't have in your data set. So how can I use? Do you intend to publish the alignment, or we just put our data and see where our diatom will be localized in the tree? I don't know if I expressed myself correctly.
I think I understand your question. The idea of how many genes to use, how much data to use is always an interesting question. We have people looking for barcodes to assign taxa, identify taxa, a lot of times in the context of environmental DNA, let alone genera and other groupings. So I don't think that question has yet been answered. What's the best number of genes, or what's the best genes to use? Myler can tell you that from his dissertation work, the group of organisms he's working on, that our lab is working on right now, is Rhopalodia and Epithemia, and their sister taxa include Cerarellas and Amphoras. It's a super interesting lineage of diatoms. And Elizabeth Ruck et al. Used 5 genes, a couple of genes from the nucleus, a couple of genes from the chloroplast, and a gene from the mitochondrion, and created a big phylogeny for that group. And Myler's using the entire chloroplast genome to create a set of relationships within that group. And they actually match up pretty well between what Elizabeth found with 5 genes and what Myler's finding with 145 genes, or approximately that. So, and there's arguments for and against using genes from different compartments. But anyway, I think that for the work that Andy did, if you ask him for the alignments for a particular group or the whole data set, he would send them to you. And you could then choose, if you're looking at 3 or 4 genes, you could pull out the 3 or 4 genes that were part of his work and create a tree, and then see where these genera that haven't been studied before fall out. For people in Brazil, you have, it's already happening, but the number of new genera that you'll find in your country will explode here in the next 10 years. Things have been so conservative, and the way that people have put species into genera there that are just so different, so different. And and the Eunochiales is a perfect place to do that work. So I think that kind of work is going to get more and more attention, particularly in places like Brazil. And I think that you'll be able to get these data from Andy and put your data to them. Whether 3 or 4 genes is enough to support the phylogeny, I think that's going to be an interesting question. There's no magic number, the thing is, we don't now know, Andrea, what is the best number of genes. And so right now it's been more is better, but I'm sure that that's not going to be the case.
Ok, André, eu acho que seu áudio está desligado. Se você quiser ligar de novo, eu só desliguei para não dar interferência.
Oh, I'm sorry, I didn't see. I was wondering if, because you speak about in future, gave us support to standard the way to classify diatoms in high groups. So in this case, your group are working to choose a set of genes, something this, because I think that everybody will need to use this new classification. So when we are getting more information and new taxas, we have to think something that everybody can use to classify in families or orders. I think that genus will not change in the class, but in the other taxonomic level can be important. We use our data to see where our position in the tree.
Yeah, so I'd say my only response to that is that we have tried to do a good job and have created this system. But as I just showed you, systems change, and we know that. So hopefully you do a good job, and there's enough information that the system can last for a while. But I assure you that when I'm dead and gone, there will be a new classification system of diatoms. And that's the way science works. I don't expect that we should, chisel this into a stone wall and say, this is the classification system of diatoms. It's, using the data we have with the theory that we've used, we hope it lasts a long time. But that's not necessarily, over time, it'll change. And hopefully with the changes, let me just say, in the old system, if you wanted to know, is there ecological information in the classification system of diatoms? Do families respond or groups of genera respond in a similar way ecologically? There was so much noise in the system because none of the evolutionary trees were monophyletic. This will give you a chance to say, we might, now that we understand that these genera are part of the same lineage, maybe their ecological preferences and tolerances are similar due to this shared history. And so you could start to see whether things within different groups of Acanthidium or whatever the genuses you're studying, that there might be a phylogenetic basis to their ecological preferences and tolerances. We've never been able to do that in the past.
Thank you.
Yeah, thank you. Appreciate it.
Okay, so it seems like we have some other hands also. And the next one is Leonardo. Leonardo, I don't know if you want to speak up.
Yes, I can speak. Do you listen to me?
Yes.
Yes. Okay, Pat. Great presentation. Thank you. I want to know, you talked about a lot of classifications of diatoms, and I want your personal opinion of which of these past classifications got each most right. Do you understand? Or they all are equally wrong, And about other thing, you said that there is no magic number for marker genes to diatom phylogeny, but I want to know if there is better marker genes, if the chloroplast gene or the ribosomal gene or the mitochondrial gene, which genes are the best for diatom classification? Thank you.
Well, let's go back to the history part first. And so, it's probably unfair of us to evaluate Agard's classification system based on data that we have in the early 2000s relative to the data he was looking at in the early 1800s. Let me just say, all of these past diatomists my reading of their work, they were all unbelievably smart, intelligent people asking super interesting questions and evaluating the data they had at hand in rational ways. It wasn't like they were just fools, coming up with stuff. They were dealing with the data at hand in a reasonable way. So I don't want to leave you with the impression that I think that they weren't smart folks. We just happen to have a lot more data and a lot of data from many different aspects of diatom biology that allow us to create a new system. Maybe so. I don't know if it's fair to say, well, this one was. I can't believe this one was. What were they thinking? I think that the folks who worked in these fields were super intelligent folks, good scientists, and dealing with the data that they had at hand. And maybe didn't have some theory that was, had been developed in, say, the second half of the 20th century. There are, so what about are there better genes than others? So here's a thing to be thinking about is you want to choose, like any question in science, you want to choose the tools that best fit the question you're asking. And if you want to know, let me say this a different way. I find it super interesting that people will use two genes, sometimes it's 18S and RBCL, to try to answer questions of diatom relationships at very high levels of taxonomic hierarchy and at species level distinctions. And I'm not sure that the same two genes have the same amount of information and noise at these different levels of hierarchy. So the first part of the answer to you, Leonardo, is that it depends on what you're asking. So if you want to know about the classification of groups related to Acanthidium and its relatives, you might choose a different set of genes than you might if you were interested in the Raphid diatoms, because there's a differential signal and noise related to these genes, and you might get very different answers if you just apply the same, particularly few genes to look at broad levels of classification. And Myler can show you lots of analyses that our lab has done when you just use one gene, like 18S, what kinds of relationships you get versus using, in our case, we're using the chloroplast genome, the mitochondrial genome, nuclear genes, and the entire genome of the symbiont to look at relationships within the group. So I guess my answer is that different questions might require different genes to answer those questions.
Okay, thank you.
Yeah. Let me just say, kind of in response to that, this is a really difficult field to be in diatoms, and particularly diatoms using molecular biology right now, because on the one hand, you have to know the diatoms, and the other hand, you really have to know a lot about bioinformatics. How different data are working, what different genes are showing you higher or less levels of variance or noise. And so more and more, just like other fields of science, I think it's really important to have strong collaborators, folks who know the biology, maybe the ecology, the taxonomy, but also people who know the informatics side of this. And so more and more, I think, being able to team up with collaborators who are on this call, who might not identify themselves as diatomists, might be a really important way to move forward.
Well, very interesting questions, huh?
Yeah.
So it looks like we have two more people with their hands raised. Next one is going to be Gustavo. Gustavo, if you want to speak, up to you. I cannot hear you. If you want to try, maybe speak. Or if that's not working, we can also use the chat for your question. I can hear it, but it's very hard to understand.
I will try to solve this problem. Pass the question to the other one, and I'll try to solve this. And then
Okay.
you call me again.
I think it's much better now. I think we can hear. Pat, can you hear?
Yep.
Fine. Okay.
Yeah. Okay. Thank you for your time, Pat. And 6 years ago, we published a paper that you reviewed, peer-reviewed, so that we placed a taxon that we proposed a new genus called Torgania, that is an eunosioid, and with a very short half system. And I'm interested most in the how do you feel the eunosioid classification right now in the light of this new classification? First, the data continue to include Eunosioides as a basal or as a sister clade of all the other raphid groups, and in relation to the rising of the Raph system evolution in the evolution of the diatoms, Eunosioides continue to be the marker of this rising, and the development of the Raph system in Eunosioides, since it is displaced from the sternum. I don't know if you remember this paper. So you discussed that the new taxa does not fit into current Peroniaceae or Eunosioideaceae family due to its morphological characters. And if you, I would know what is I would know what is your opinion concerning this split of Eunosioideaceae in other families, and mainly the genus Eunosioides, which is extremely big with a large number of species.
Yeah, thanks. I do remember the paper, and it's that group that I had in mind when I said that I think there's a tremendous amount of work to be done in Brazil, this on diatoms generally, but this group in particular. So the origin and evolution of the raphid diatoms is an unbelievably important part of the diatom tree of life. From lots of different perspectives, ecologically, in terms of evolutionary rates. There's been a paper published that suggested that raphid diatoms are evolving 2 to 3 times faster than other groups of diatoms. And there's all kinds of things that fall out of that. And yet, our understanding of what happened at the earliest stages of raphid diatom evolution are still pretty unknown. And the Eunosioides or Eunosioideaceae is an important junction in the tree of life of raphid diatoms. And so, if taxon richness is an indication of diversity and evolutionary activity, the Amazonian part of Brazil is a place that one would want to go because of all the myriad of forms and morphological variability, which we would expect to be expressed molecularly as well. So I just think that one time I was trying to put together an international team of people to study this idea of what was the earliest Rhaphid diatoms like, and the importance of understanding that early evolution. And if I was going to do that work, it would be in Brazil. You guys are they're geographically so positioned to do that work. And for the genus Eunotia, I'll just say that any genus that has 2,500 taxa assigned to it is probably not a natural group of organisms. And so I think that, it's time to separate out that genus into natural groups. And I don't believe that the only thing that holds the groups together in Eunotia is that they're not Actinella, they're not Turgeneia, they're not all these other things. And that's not a good way to group things, is by the lack of a feature. And so I think genus Eunotia is, should be the first on the list, maybe after Pinularia, as the genus in freshwaters anyway, that needs a revision, systematic revision. It's prime for that activity.
Okay, just one more question. All the half systems, including the Eunotia type half, do you think they all are homologous, including the Eunotias, or development of the half system, the ontogeny of this half system? And the second, in terms of biogeography, Amazonia has been thought to be a hotspot for this group. The book by Lange Bertalot, Metzeltin, the 2 volumes of tropical diatoms, they revisited the hosted samples. But you has been working with the other tropical places in Asia, and also found new species related to the Eunotiaceae. Do you think that further sampling in these tropical countries will prove that they are also hotspot for this group, or do you think that the Amazonic region is more suitable in the history, in the evolutionary history of the group?
So the first part of your question is the Rhaphid system homologous amongst all the members that have Rhaphid systems? And right now, there's no data that I know that suggests that the Rhaphid system evolved twice. Now, actually, if you look at that little diagram by Simonsen from his 1979 paper, it suggests that it could have been evolved twice. But molecular data and morphological data that have been analyzed to date suggest the Rhaphid system evolved once. So my answer would be that it looks like the Rhaphid is homologous amongst all members who possess it. With respect to biogeography, I would say that if I was going to do the work that we just talked about on the Eunotiaceae, Amazonia would be the first place I would go. But the second place I would go probably would be West Africa. And there are lots of asset habitats there. If you go back and look at the divergence time of the Eunotiaceae, it might align with the separation between South America and Africa. And I think that from a biogeographic point of view, there might be a lot of information contained in looking at sister taxon relationships between Amazonia and West Africa. So while Asia has a lot of interesting taxa, and you made reference to Cynoporonia, which, by the way, is a Eunotioid diatom, but it has birapid frustules, monorapid frustules, and arapid frustules that it produces. Yeah, that's what I'm saying, Olivia. And yeah, it's crazy. So it's an interesting place. And I'm told that that people have found that in Venezuela, but I haven't seen, it's not been published as far as I know, but I'm told that that's been, that genus may be in Venezuela as well. But I really think that, you know, in addition to Amazonia, another understudied area would be West Africa. It might have very interesting biogeographic implications.
I told that it was the first question, but I want to one more.
Sure, it's a chat. This is the way chats work.
Yeah, ideas appear in my head. But I remember what Kaoli asked about the diatoms being included into the broader group that is heterocontophyta. And what do you think about the system or the approach that the researchers of angiosperms like APG, where they are prioritizing the organization of the natural groups more than if it what Linnean group they belong. So they are trying to organize molecular data and achieve truly monophyletic groups. And this is more important than if it is a class, if it's an order where the disclades, how disclades are being placed. What do you think that this approach it could have some application in your system, in your new system?
Yeah, so there's been, you know, for quite some time, actually, people have tried to wrestle with the challenge of how restrictive Linnean categories are to express the diversity of life. And I've you know, I also work with something called diatom base, and in that system, the number of categories are actually they're numerous. You know, you can have a supra family, and there's a whole bunch of other prefixes you could attach to these Linnean categories to create a lot more categories. I do think that names are super important. It's you know, you don't want to say, look at branch number 49.27 and start to create a classification system like that. Although people have tried, you know, there are these phylogenetic classification systems that are devoid of names, and they just look at branching position. I think there's really a lot to be said for names, and it connects you to the literature. It provides a lot of information that is not easily dealt with by numbers or. So I think that we probably could expand the Linnean system. There's a lot of category names. It's just going to end up with a lot of descriptions. We tried to avoid that in this paper. We tried to not do all these other subcategories. And that's why some of the classification categories might seem that like there's a lumping of things under certain names, because we just told ourselves we were going to try to stay within the strict Linnean system. But I'm not a big fan of saying, you know, it's give a particular branch a number or something like that. Thanks.
So I think that it will be useful to in the future use more of the family approach in diatoms to likewise they are doing with the angiosperms, working if you have the families, monophyletic families, and it would be a good way to refer to the diatoms, to the major groups of diatoms or class. What do you think, or orders, which would be better in the case of diatoms?
Yeah, I'm confident that, so we have been for almost 200 years, maybe more than 200 years, focused at the levels of genus and species. And I think that more and more, the higher level of classification for diatoms is going to be more and more important. And in that we will see more and more publications focus at the level of family and order. I'm sure of that.
Yeah.
Okay, thank you very much, and thank you again.
Thank you.
Thank you, Gustavo. Looks like we have a lot of questions, just like science is. But the more questions we do, the more questions we we have. And speaking of questions, so the next one is coming from Wellington. So up to you, Wellington.
Sorry, Patrick, I will use your moderator and I will make the question in Portuguese. Okay?
Sure.
Actually, I have two questions. Melhor, se puder passar para ele, para a gente, vou complicar sua vida aí. Essa é uma das filogenias mais completas que a gente está tendo ultimamente de diatomáceas. Então, a primeira questão é relacionada ao seguinte: Medlin e outros autores, quando começaram a fazer suas filogenias, fizeram várias inferências sobre a questão da origem das diatomáceas e mesmo de como elas chegaram no ambiente dulceaquícola ou como voltaram para o ambiente marinho, saíram do ambiente marinho e assim por diante. Então foram várias conquistas, né? Como que essa filogenia também que eles apresentam, que é bastante completa, não totalmente completa, mas assim, a que a gente tem de maior representatividade atual. Então, como que ele vê essa questão da diversidade de diatomáceas nesses diferentes ambientes? Eles já começaram a pensar em algo a respeito disso, tá? Então, dos diferentes ambientes, marinhos e principalmente marinhos e de água doce. E a segunda questão, agora eu vou puxar a sardinha para o lado da presidência da rede, ele mencionou o papel do Brasil na questão dos estudos das diatomáceas, dos gêneros, da diversidade que a gente tem que ainda não foi explorada. Então, a pergunta que eu queria também, a segunda pergunta, era a opinião dele de como a nossa rede, né, de como esse tipo de trabalho poderia contribuir para esse preenchimento de lacunas na própria classificação. Então, como que o nosso trabalho em rede poderia atuar para a gente contribuir com esses estudos e conhecimento da diversidade de diatomáceas nesse nível de classificação?
Okay, so it looks like we have two different questions, and I'll try to summarize the idea for that. So first is Wellington mentioned that Medlin and some other authors, they have inferred multiple different hypotheses about the origin of the diatoms, and also this more focus on these transitions between freshwater and marine and back and forward actually for the diatoms. So the first question is related to how your phylogeny or the phylogeny that you're presenting in your paper is related to the diversity in these different habitats. Is there a relationship between the habitats, and have you investigated this biodiversity or the habitats that diatoms have if they transition to the different places, and if you actually see any patterns correlating phylogenetic signals and also the habitats? And then the second question, you also mentioned that Brazil will be a hotspot, it looks like we do have like the gold in our hands to study these organisms and all of this. How can our network, how can our this Brazilian, how our network can contribute to future classifications, and also how can we keep up with the pace with this taxonomical updates and all this new classifications of diatoms and all of that.
Wow. Okay. Well, I'll see if. Well, let me say that there's been if I understand the first question, the sampling of the taxa for the phylogeny that Alverson et al. Did is very broad. It's, marine, estuarine, fresh waters, lots of groups from lots of different types of places. Yeah, but if the question is related to this idea that there's a Rubicon between marine and fresh water, I don't know if that's exactly what Wellington was thinking about, but if there's been this proposal. It started out with Sims and Round, and later by Dave Mann, that diatom genera are either freshwater or marine, and that crossing that Rubicon, crossing that salinity threshold, is very difficult for diatoms. I'd say that most data there's a resounding no to that idea. That's been identified many different ways with many different groups. And so while a genus might have most of there's two issues. So and this can come from our recent research on Rhopalodia. Rhopalodia is a genus that has marine and freshwater representatives. And in Rhopalodia, there's about 250 described taxa. It's clear that people have lumped many things into Rhopalodia, and that there are marine lineages of Rhopalodia that never get into freshwater, and there are freshwater lineages of things called Rhopalodia that never get into marine systems. But using just the term Rhopalodia confuses the entire situation. And so we would be best to, in my view, to split many of these things out into separate genera, and then we could have a much better conversation about, is it specifically marine or specifically freshwater. Under this big lumped group called Rhopalodia, it's difficult to have that conversation. But I'll just say, in Amphora, Nitsia, many groups, Thalassiocyra, the idea that things can, that two taxa that are sisters, one can be in marine and one can be in fresh water, is very well established. So I would say if that's the kind of the basis of Wellington's first question, I'd say that it looks like many groups can easily cross this Rubicon of salinity. With respect to the position of Brazilian diatomists in terms of their natural endowment, the wonders that they have been given in their ecological habitats and their contributions to that, I think that I can't express enough the need to continue to go to nature and collect and observe and study natural communities. Of course, you might want to get them into culture, but nature has many, many things for us to still to describe and discover. And I would encourage people to keep a field element to their research programs. I would also suggest that while we can have a wonderful chat here about diatoms, that it's important to get out and talk to people from other disciplines, get different ideas about how to think about research programs and questions, and to collaborate with people outside our disciplines. And I would include informatics, people who specialize in informatics, to have a particularly important role in that in the future. So, I think I think it's not a good idea to just rely on sequence data and NCBI and cultures from culture collections to do your work. I think that there's too many treasures still to be discovered. So I think an aggressive field program and collaboration is a really important way. And keeping up with here's a challenge for all diatomists, I think. We're still in a discovery period of our discipline. We've only been seeing diatoms since 1703. People who study birds have been seeing birds since there have been people and creating classification systems of birds for 1 million years. And, if you give diatomists 1 million years, we will figure out all the taxa and all their relationships like ornithologists have had. But we've only been studying for 250 years. So just keep in mind that we're still in a very early stage of our discipline, and we still have a lot to learn about them. So making forward progress is important, but we just have to be prepared that new insights, whether that comes from new habitats, new places, new tools, is going to be the norm, and that will then require adjustments, including things like taxonomy along the way. So don't be frustrated by it. I think it's just, you have to embrace it. And luckily we live in the internet age, and we're still not, I would imagine many people on this call didn't live through what I lived through as a student, where you physically sent a card through the postal service to ask somebody for a physical reprint of their science, and 3 months later you might get it. So you live in while there's a tremendous amount of change, you also have tools that a previous generation did not.
Perfect. Thank you so much.
Let me just say, before we,
Sure.
I just want to say, I can't see everybody that's here, but it's great to see Kociolek and Margaret and other people here that I've had the pleasure to interact with in Brazil. And it sure would be nice to go back to Brazil sometime. So please, if you think of a thing to collaborate on or work together on, I'd be love to do that. Great to see some of my colleagues again.
Oh yeah, maybe we can arrange that travel patch and go collect in Brazil.
That sounds like a good idea.
Yes.
It looks like we also have a question from the chat from Gisele Marquardt. Well, first she thanks you for the lecture, and then the question is, given the expected increase in new genera in Brazil specifically, especially from fossil records, how do we avoid inflating artificial taxa when their monophyly cannot be tested?
Well, I think my answer to that is, so let's think, so what are the options? You could do nothing, right? You could just say, wow, that's interesting. I can't tell it's monophyletic or not, so I'm not going to deal with it. That's one option. And that's not a good option in my mind. Another option is to say, I'm going to describe it. It doesn't look like it matches anything else that I can see given the tools that I have. I'm going to describe it, and it is going to be the job of future generations with new tools and new ideas to sort it out. Now, the thing is, if you don't describe it, no one can sort it out. No one can see the taxon. If you describe it and it turns out to be non-monophyletic, okay, well that's not the way we wanted to do it, but maybe we'll find out in the future that we need to sync it into a different genus or whatever the taxonomic or nomenclatural activity is. So I've always been My philosophy is, when in doubt, split it out. And then somebody else can later come and fix it. But if I hide it by lumping it into something else, no one can see that taxon. And so if I'm going to err as a scientist, I want to err on the side of making too many things that need to be sorted out later, but exposing that taxon and its features to the scientific community for further study. If I just hide it or I don't do anything with it, it's lost. It's basically we don't even just. So I think that's a worse way to go. Neither one's great, but if you're going to be wrong, I'd like to err on the side of sorting it out.
Perfect. Well, it's so unfortunate that we only have about 10 minutes. We have probably time for a few more questions. And someone already said that we should be thinking about our chat number 2, or the second part of our chat in the future as well. And well, Davia, which is right there, suggested, well, thank you for your presentation as well. And then I'm pretty sure that I can say this from all the diatom people in Brazil, that we are super excited, and these new things that generate all of these questions and that is enthusiastic is something that it gives us more energy to investigate all of that. And yes, any more questions?
Let me say that I'm honored to have been the first one to participate in your chat, and thank you again for that wonderful invitation. And of course, if I can help any of you in any way, please don't hesitate to drop me a line. If you need some literature or something like that, I'll be super helpful, try to be helpful to you. So just keep that in mind as well. Myler can tell you that our lab is overflowing with literature, and gets in the way of people space. But if I can be helpful to you in any way, please let me know.
Yeah, I by the way, I also put myself out there. If people want to reach out, please feel free, either in English or Portuguese or whatever works. We're here to make this connection and to make our network work. It is already working.
Yeah. Thanks again, folks. I really appreciate it.
Mais alguma questão, pessoal? Another questions? I would like again to thank Patrick for its availability. It's very important, this kind of chat, this kind of change, because sometimes we need, connected people with the big reference that I think that you are. And I think that it's very important. Sometimes I make some this kind of initiative to connect the people and make the people see and talk with the great authors that publish about diatoms. Then again, I thank you very, very much, because I think that this contributes so much with our research and with our initiatives. Thank you so much.
My pleasure. Thank you. Bye,
E
everybody.
pessoal,
Good to see you again.
E pessoal, eu agradeço também imensamente a participação de todos. Agradeço o Maylor, que foi super atencioso e auxiliou imensamente a gente quebrar essas barreiras aí, até mesmo fazer os contatos. Então, muito obrigado, pessoal. E se não tivermos mais nada, ou a gente ainda tem 7 minutos, eu já queria falar que esse já foi o nosso primeiro Papo Diatomáceas, e a gente abriu com chave de ouro, fala sério, né?
Well, I would also like to extend my thanks, Pat, to be available and to share your work and your expertise with us. It is my great pleasure to do this, and I'm happy to see some familiar faces as well. And for those who don't know me, reach out. We're here.
Great. Perfect. Thank you.
Thank you, everyone.
Só para informar a informação final, nós gravamos essa reunião e, posteriormente, já que o Patrick autorizou, nós vamos colocar, então, no nosso YouTube, tá, pessoal? Para quem depois quiser reassistir, verificar. Então, agradeço a todos e depois a gente vai disponibilizar o link dessa apresentação para quem não pôde ficar atendendo. Eu já fiz alguns prints aqui, mas a gente podia tentar fazer mais um print, né? Eu vi rostinhos novos.
Would you mind if we do like a quick picture of everyone that's
Yeah, that'd be great. Yeah, let's do it. Good idea. As long as everybody share it out.
Okay.
Come on, everybody, get on. Turn on the video.
Okay, great. I make a lot of pictures here. Okay.
Wow, we got
Thanks so much. We have another people. Okay.
Great.
We have a lot of graduation students here, and this make me very happy because this will be the next generation of diatomologists.
Absolutely.
Thank you so much, people, and
thank you.
Meet you all in the next Papo Diatomáceas. Thank you so much.
Okay.
See you all next time. Bye.
Diatomácea.